My name is Eve Schneider, and I'm an assistant professor in the University of Kentucky Biology Department. I never in my wildest dreams thought that I would actually end up as a scientist. Science is amazing. It's a privilege to have this job. It's a privilege to get to be a teacher, to come to a place like Kentucky and be in a department that specializes in evolutionary biology. Getting the Beckman Foundation funding was an incredible opportunity because, first of all, just the validation that somebody out there thought the work we were doing was good and that I could be part of this kind of elite group, or what they call the Beckman family. I went to art school for undergrad, and I actually majored in sculpture. So I started making a lot of art about kind of the politics of psychiatry and getting more and more interested in kind of the medical model of psychiatry and, sort of socio-political aspects of that. And realizing that as I was making this artwork about that stuff, I had no idea what I was talking about. So I started taking psychology classes and kind of gradually veering over to neuroscience. By the time I got my art degree, I basically had enough credits to have a second major in psychology. I applied to a bunch of grad schools, and I got into Princeton. During grad school, I was TAing a course in cellular and systems neuroscience. The professor who taught the course, at the end of the course, he said, "You know what, you can join my lab." And then it was crunch time. I mean, the stuff we did was really difficult. I did not have a computational background. I really had to hustle to get up to speed. So part of the challenge was trying to figure out what exactly I was going to do my PhD on. At some point, I started recording from cells in what are called the deep cerebellar nuclei. Cerebellar activity influencing the development of other brain regions, so parts of cortex that are important for social behavior and that type of thing. As much as the cerebellum is not understood, the cerebellar nuclei are really not understood. I collaborated with a postdoc who was doing 2-photon imaging, and I said, "Hey, I'll make the slices. Can we just get one of these? Can we get some of these cells under the microscope and see, do they have calcium signals?" And we did, and they had tremendous calcium signals in their dendrites. They were like 200% change in fluorescence. We took the data into Sam's office. We're like, Sam, look at this. And he was like, that was the moment I knew you were going to get a PhD. Yeah, by the end of grad school, I was looking around for postdoc opportunities. There was a lab studying, it was actually advertised primarily as studying mammalian hibernation. I responded to this ad in Nature Jobs to work with Slav Bagriantsev and Elena Gracheva. I told them about my research. They were telling me about projects that they were going to work on in their lab. And then Slav mentioned, we have this other kind of side project that we're also interested in. It's looking at the sense of touch, and we're going to use birds that forage using touch. And so I joined the lab, and basically to work on both thermosensation in mammalian hibernators and also the sense of touch in tactile foraging birds. When you're trying to understand the molecular basis of something, you need some fresh tissue from the animals you're working with. The types of corpuscles that the duck bill skin has are actually very similar to what we have in our palms and fingertips. Anything that you could study in an adult duck, you can also study in a duck embryo. A lot of the work I did with my Arnold O. Beckman postdoctoral funding was to show that the duck tactile system is pretty much ready to go before they even hatch. The real advantage of working with duck embryos is you can manipulate them genetically. So a lot of what we're doing in my lab now is actually trying to image cells that sense touch in the duck trigeminal ganglia and also in the skin, and actually get more sense for, what are the properties of these signals. What's interesting about the duck is that their bill is incredibly sensitive, but the rest of their body, not so much. We recorded from a bunch of duck sensory neurons and found that they were much more sensitive to force than mouse neurons, and also that they expressed a ton of this piezo-2 channel. And ultimately, with the support of the Beckman Foundation, we ended up cloning the piezo-2 channel from ducks and showing that its behavior is actually very similar to what's been seen in other vertebrates. So these channels open very quickly, and then they also, in response to a sustained force, they also close very quickly before the force ends. My husband is a lawyer, and he vacillates between being incredibly sympathetic and understanding and also making tons of jokes about science. My lab website is called quacklab.net. He was like, hey, you work on ducks, your lab website should be called Quacklab. One of the Beckman principles is don't take yourself too seriously. And that one I can say I embrace pretty wholeheartedly. Of course we strive for perfection. You know, there are things that we really aspire to as scientists, but we're also humans. You know, we're scientists, we're moms, we're spouses, and it can be pretty challenging to really bring your best self to all of those things. As a working mother and a scientist, some of the best role models I have are Beckman Young investigators who manage to seemingly effortlessly balance their amazing scientific achievement with being good parents. Toward the end of my postdoc, I had my daughter Rita. You know, I was able to have maternity leave as a Beckman Fellow, not have that questioned, you know, kind of have a little more independence and freedom than I may have been granted otherwise. I was extremely lucky that during my first year on the job market, one of the places I interviewed was University of Kentucky, and ultimately was hired. This department is pretty amazing. First of all, it's a biology department. Such a broad range of specializations. It's really a rich environment for collaboration and for inspiration, because we all talk to each other, share ideas, and you end up thinking about doing experiments that you would never have considered. So it's been a year of tremendous change. I mean, going from being a postdoc to being a PI, having my daughter, becoming a mother, and without the support of the Beckman Foundation, both financially and kind of morally, in terms of the set of connections, the Beckman principles, I'm not sure that I would have withstood some of this change as well as I have.